graduate student in Scott Hodges' lab. I'm not sure about the microphone. I'll probably paste back and forth. Okay. Rapid diversification of lineages provides ample opportunity for incomplete lineage sorting and progression to move alleles across species boundaries. This has the potential to add genetic variation during adaptive radiations, and it certainly complicates population genetics and inference about cognitive mutations. So I'm going to share some results that I have regarding color shifts in Australasia that make me think these complicated processes of introgression or the maintenance of ancestral polymorphism have been integral to the adaptive radiation in North America. Columbine has a great diversity of coral morphologies, and these change within a suite of correlated traits associated with pollinator shifts. I'm specifically looking at color, which ranges from an ancestral type blue seen on this bumblebee-pollinated narrow endemic from Colorado, to independent shifts to hummingbird pollination, where we see a shift from the blue anthocyan to the red anthocyan and the gain of floral carotenoids, and then at least 5 independent losses of floral anthocyanins associated with a shift to hummingbird pollination. And whether the flower ends up being yellow or white, like the cerulean, depends on the underlying floral carotenoids. So along with all of that variation between species, there's variation within species as well. For example, there's white cerulean, Rocky Mountain columbine, was in a population along with numerous blue flowered individuals. So what I wanted to do is get a handle on the mechanistic basis of this variation within and between species. And the approach that I took was RNA sequencing. And my advisor Scott and I sampled extensively in natural populations. We had to rely on field-collected seed or horticultural lines in a few instances. But by and large, we sampled tissue in the field from wild individuals. I ended up with a total of 76 transcriptomes, and each of those consisted of potentially multiple individuals that were anonymously pooled. So each transcriptome consisted of anywhere from 1 to 15 individuals anonymously. Now, when doing a differential expression test in a phylogenetic context like this, I was really worried about dealing with the fact that these are not really independent replicates. So to account for phylogeny, I first identified SNPs transcriptome-wide. Then I performed a principal component analysis of those SNPs to generate PC axes that I could use as covariates in my differential expression model. And it was very gratifying to see some well-supported phylogenetic relationships where species blue Eurasian species cluster over here, North America over here, ancestral type North American are distinct from this derived clade or group, I guess. And there's a whole bunch of color variation just mashed in there. You can't really see it. It's all mashed on top of each other. So I used principal component axes as covariates. Cartoon of the model here. Color plus this phylogenetic background allows me to test for differential expression. Fit that model and then look at specific contrasts relating to the shift of interest. So the first shift I looked at was from purple and blue to red. And the shift from blue to red. And what I'm looking for is consistent changes in gene expression or sequence associated with becoming red. And rather than show you the whole transcriptome data, and there are thousands of differentially expressed genes that are significant after accounting for multiple concentrating on anthocyanin and biosynthetic candidate genes that I identified. And when we look at these two comparisons, purple, red, blue, red, there are 3 candidate genes that stand out as having highest significance in a very large fold change. One of these is a positive regulator, an R2-R3 mid-transcription factor with high homology for a gene called PH4 that acidifies or leads to acidification of a vapor. That's really important for anthocyanins because in an acidic environment they shift towards red, and this PH4 homolog is at high expression in the red flowers. The other gene that's really interesting is F3'5'H. This is at a branch point in the anthocyanin biosynthetic pathway, pushes substrate down the delphinitin branch and leads to a blue flower. It's expressed at very low levels in red flowers, high levels in blue and purple. This third gene is a bit of a mystery. It could potentially be related to modifying the anthocyanin once it's been produced, which could alter its color, it could stabilize it. It's a member of a large family, and whether it's actually involved or not is unclear. Of these 3 genes that I could identify expression differences in, I was very limited in the ability to look at sequence differentiation. Only F3'5'H had enough sequence where I could actually do anything with the differentiation. Again, this is a principal components analysis, but instead of a whole transcriptome, it's just the locus of interest. And the really striking thing to me is that there's red down here, there's red up here. This is canadensis, an independent shift to red, and all of these are derived from a single shift to red. So both of these have low expression of F3'5'H, but they have high sequence differentiation, suggesting a different haplotype carrying different mutation. So the inference from this plot is that this is an independent loss of expression for the cis-regulatory change. Now I didn't have expression differences for DFR, but there's strong sequence differences for DFR. DFR is an enzyme that in other systems is known to evolve substrate specificity for blue versus red anthocyanins. So we see that reds are clustering. Canadensis is again separate from the other reds, but separate from the blues. So again, this looks like independent mutations leading to specialization of an enzyme. So the other shifts of interest are the loss of anthocyanins. I compared blue to absent. This includes yellow and white. And here, I'm plotting the whole transcriptome results. The magenta circles are anthocyanin candidates. And you can see that there is an incredibly strong pattern of coordinated changes in gene expression. Most of the pathway is upregulated in the blue flowers relative to the lost flowers, the anthocyanin minus. This really looks like a transregulatory change. Red versus absent, same exact phenomenon. We're seeing dramatic changes across the entire pathway, suggesting a transregulatory element. Another way to look at this is, again, with a heat map. The columns are the individual samples, and these gray species, these are polymorphic. So these are present-absent polymorphic species, and I'm just including the absent species here. Blue indicates that expression is higher in blue flowers. Grays and black indicates expression is higher in the absent. So again, we're seeing this coordinated change in expression, but it's not nearly as strong in the polymorphic species. So this suggests that the loss of anthocyanins is consistent when the species is fixed for loss, inconsistent when it's polymorphic. Same exact pattern when we compare absent to red. Okay, so we have this hypothesis that the change from present to absent is caused by a transregulatory element. I have 3 candidates that I identified based on a number of approaches. And these 3 candidates are on chromosome 6, they're tandem duplicates, high sequence similarity. And what I'm looking for here is clustering of present versus absent. That would be nice. That would indicate that there's something going on there. I don't see it for 1433, we'll call it. 1434, colors are all mashed up. 1436, there's a nice separation of red versus blue, but we're not getting separation between present and absent. That was a little disheartening, but this is only SNP data. I'm not looking at indels here. So there's a strong potential for indel polymorphisms to cause branch shifts or loss of function mutation. So I did this the hard way and just scanned the alignment by eye. A number of very interesting branch shift mutations. So here I've put anthocyanin minus taxa that are fixed up at the top. Polymorphic in the middle here, fixed for present down here. Skinneri is kind of cool. This is a species that makes anthocyanins in its petals, but it has green sepals lacking anthocyanins. So the gray box indicates that that species is polymorphic for the mutation of this position. Black indicates that it's fixed. So we see that every one of the anthocyanin minus species has something somewhere going on that is going to mess up this transcription factor. Very little fixation. There's substantial genetic heterogeneity here. And in, for example, in flavescens, it's really quite remarkable. These are two different polymorphisms that are out of phase from each other. They're on different haplotypes. Same for these two down here. This is too far for a single read to map across it, so I don't know the phase here. I found that really surprising. So much genetic variation potentially causing the same phenotype, segregating within populations, across species boundaries. It's super cool. It's a mess, but it's cool. With that,